Okay, shall we get started?
Yeah, okay. So I think, I read through the goals of the session and what we're supposed to power through. And I think that's going to be, a little bit impossible. So everyone is going to have to keep the time, both in, scheduled 5-minute slots as well as questions and answers. I'll try and keep the time, but I'll welcome everyone. I mean, we're really switching gears here from talking about atlases to build an atlas, and there's so much work yet to do, even though a lot of data and a lot of coordination has already begun. So I think the goal of us to be here is, it's a little bit weird to mix entirely different bio-networks, but again, I think the goal is to learn from one another. Part of the goal of getting the human cell atlas rather than the lung cell atlas or the breast cell atlas or the pancreas cell atlas is to learn from each other. And also, there's a lot of both connections, commonalities, and unique differences. And I really believe in comparative biology. So I think the session starts with 5 minutes from each bionetwork. I mean, right now, one of the most critical things that the HCA is really doing is putting together a roadmap. And the atlas, I mean, of course, this is just, the first version, first draft. We're going to find all our flaws. I'm very good at being critical. Still, we'll do a much better job with all the money being invested if our roadmap is good. So I think our first presenter is liver.
Yep. Bruce Arnold.
Bruce, and again, I really have to stress, I know you have a lot of smart things to say always, but I'm going to be, brutal in timekeeping, 5 minutes, because we're so many bionetworks, and I got an insane list to cover. You should have a second, there's the second microphone, okay. So describe your roadmap for the liver.
Okay, so the liver, I think, was one of the very first seed networks that was established, and so that's under Mass General, and the it's a really large group, and there's I think a lot of data that's coming together. There's still some parts that are not some data sets that are not quite in there, but it's going to be of the order of 30. Different, I have my full list that's written out, but it's a mixture of cell-seq and nuke-seq and multiple spatial genomics technologies, including MERSEq and let's see, then also and ATAC-seq. So there'll be healthy adults, children, and then there's now a pediatric liver atlas that's added in, and the liver is it's a tremendous organ that it converts from producing blood cells in the fetal stage to producing blood proteins in the adult stage, and the number of functions for digestion and anti-inflammation and management of hemostasis and management of nutrition and cycling. It's just, there's a tremendous amount of biology in the liver, and so being able to sample variations and all that is beginning to be within the scope of this data set. And now with Gary Baders leading the pediatric developmental component of the liver that's been added in, and there's some even more exciting work in there. Gary's developed a new approach to electron micrographic imagery constitution and 3D visualization. We saw some of that, the 3D visualization of cell-cell interfaces and kind of the very fine structure of niches and that are the basis of cell-cell interaction. All of that's to be cataloged in the liver context. And another thing that's not in this so much is it's going to have to be inferred is variations in feeding state. So the liver is, has a very high circadian regulatory activity, and it's not clear that we're sampling deep enough to go into that. But now with diversity and that is another variable in that, and then feeding status and
So how did you plan the atlas to try and I didn't get a sense for scope, number of samples, number of cells. How did you come together to actually do smart experimental design? What does your roadmap look like? And again, I'm really happy that a lot of cool, looks into the spatial and epigenetic angle is coming in. I think they're critical. I'm a big fan. That interests me personally most. But, we want the first version to be, a reasonably comprehensive atlas that's reasonably designed with coordinated design. So how are you coming together on the roadmap for the bulk of it?
There's enough expertise and there's tremendous expertise there such that some problems were evident from the beginning. So stellate cells, as is common with adult organs, these mesenchymal cells that have a lot of collagen, they weren't released well into cell-seq-based methods. So then there was a commitment to the having a high amount of nuke-seq data that would be part of it, so that you could do comparative studies, and then similarly, neutrophils are one of the larger immune cell types of the liver, and they decided that really there should be a similar commitment to well-seq.
You can wrap up because I was told to keep it strict 5 minutes. So.
Okay, so yeah, I think multimodalities and integration of annotations, reference signatures, but being able to now have a base reference for understanding cholangitis and fatty liver disease and liver fibrosis, that sort of framework should be there in the variation in states and that can be compared to.
Okay, so thank you. Now I invite Lung up, and we'll have much more time for discussion. But this is just like your 5-minute overview pitch. Imagine that I get to decide which of the 4 atlases in the room to fund. So, pitch why your roadmap is the best.
Okay, so hello everybody. I'm Pascal Barbry from Nice, and I'm speaking for the Lung map. So don't be afraid, I will not give you a new presentation of what Fabien has already showed, and then Malte. So you are aware of what has been done till now with the Lung. What will be the next iteration that we have in mind at the moment? Well, first of all, there are discussions with a project like a Lung map to create a common reference. I think that it is very important to create a common view that can be shared not only with the community of people interested by single cell, but that becomes also a tool that will be available for all the people interested into the Lung, and that will use it to map their specific studies. And so I think that it is very important that we agree all in the community, in the small single cell community, about what we are presenting, and that we share this data to everybody. So it implies that we are working together, and there will be probably a new iteration of the atlas that will come with that. With that, I think that it will be interesting to get a better resolution at the anatomical location. And this is possible because in our case we are sampling with bronchoscopies, and it's possible to get very precise sampling. What is also very interesting in the model is that we can have access, this is at least possible in Europe, to LC individuals that we can sample like that. And that makes also something very interesting because we have the real reference that is not a normal area of something that is pathological, a tissue that is otherwise pathological. This is fully LC control. So this will be important to improve the resolution, to have more variation, as we already alluded, in age, ancestry, environment factors. And then all the other modalities regarding the spatial or multi-omics are also of interest, and they were also very well described before. I think that the last point that deserves some interest is there are two things. The first thing is about the link with pathologies. It will be very important to develop the study now, not only on the normal and healthy tissue, but also to look at pathologies. And I would like to advocate a little bit the biological view of it, meaning that it will be important to keep in mind the correct experimental design to have the good control and the good system to make the comparison and not...
Yeah, if I can interject, actually one of the things that I find interesting about the lung is that you guys rebelled from early on, and as the HCA and CZI and all of them were saying, no disease tissue, the lung said, no way, disease is in there from day one. So why and how's that worked for you? Because, you know, you're talking about pathology, and disease is one of the, you know, central things in the Healthy Lung Cell Atlas as well. So if you could talk about that for 1 minute.
Yeah, well, I think that it is a good description of what's going on, and it's also, yeah, there is also a last point that is of importance. It's regarding the developmental status, and I think that in the context of the lung, it is particularly important, and we will need to look more carefully at what's going on during the fetal stages. And yeah, just the last point that is important is to go to couple what is done in vivo with what we can do with organoids and with models, either the animals or the in vitro models. And I think that it is very, very important to have this coupling because it will demonstrate the importance of what we are really detecting by the single cell and all these approaches.
Okay, thanks. We'll have lots of time for discussion, even though we have way too many targeted questions. They gave me 50 minutes for about 3 hours worth of material. Reproduction is next.
Yes, on behalf of Reproduction, we have Rosa Vento and Andrea Jones joining via Zoom, and combined, they should spend 5 minutes updating us.
All right, let's have,
Yeah.
Are they good on the screen? Great. Okay, I can see, oh.
And Gary
Thank you.
Bader
Can you
joined us
hear
on
me?
Zoom as well, on behalf of Deliver for future
I'm
questions.
going to tip this over, so.
Rosa, go ahead.
Can you hear me? Yes?
Yes.
Yes.
Okay, good. So yeah, I am Rosa Bento at the Wellcome Sanger Institute. So we have started on the Atlas of the Female Reproductive System, and we have been focusing on the ovaries, fallopian tubes, and uterus. And we feel these are essential for reproductive success and generally in women's health, but remain poorly covered in early HCA efforts. And one of the main challenges for this is the nature, the dynamic nature of the tissue, because during development there is the cell fate determination is accompanied by sex specification, and in adulthood the tissue responds to the fluctuation sex hormones. So we are generating tissue atlases of reproductive tissues from both development and adult tissues, and including the uterus, the fallopian tubes, and the ovaries. And we are profiling the tissues using single-cell genomics and also spatial transcriptomics and imaging methods. So in adulthood, we are covering both non-pregnant and also pregnant women. And currently, we are also generating data sets of pediatric reproductive tissues and also covering postmenopausal samples. And we feel this is really essential to cover the whole heterogeneity of the reproductive tissue. And it's also important that we are generating data from also in other efforts from hundreds of individuals and also including different ethnicities. And we feel this is important for reproductive because it's known that genetics and ancestry play an important role, but the association is really not studied. Other ongoing efforts include the integration of all dataset using bioinformatics tools. And again, this is important to understand how the tissues talk to each other and how all this crosstalk is changing across the lifespan to understand the reproductive tissue as a whole. So we highlight 3 challenges. The first one is the variability within individuals, all this tissue regeneration with the
That's
hormones.
a challenge and a blessing.
The definition of a healthy tissue, just as a friendly reminder that 40% of women report in ecological conditions, and yet we know very little about this, so how we define the health. Reference, then the tissue accessibility, as often we are limited to kind of pathological tissues such as infertility, and also some of the developmental windows that are not accessible, such as the implantation. And then the spatial variation that is often lost in biopsies. So to overcome all of this, we are also profiling in vitro models of the reproductive tissues, and this is allowing us to study the developmental windows that are not accessible in humans, this black box of pregnancy, and also to, in a way, correct for this inter-individual variability, for example, when we cover different stages of the endometrial regeneration. And also the in vitro models are kind of letting us to remove the environmental effects, saying these as, for example, the fluctuation of hormones. And the other thing that we are moving as well is towards the multimodality, including 3D atlases, and this will give us a better resolution of the cells and tissues and deep understanding of the function of cells.
So I have a bit of
we are planning to do that in health and disease and multiple samples with different ancestries. And that's, yeah,
so I have some questions about technologies.
Mention their updates on the ovary.
So I have some questions about technologies, because last time I met you guys, which was very ancient ago, pre-COVID, this network was really into drop-seq because it was cheaper and you can get more cells. So are you still, what technologies are you using both for the bulk of your RNA-seq, and you're talking about 3D mapping, so what technology are you using for that?
Yeah, so we are not using drop-seq actually, we are mostly using 10x genomics.
Good.
And we, in the majority of the studies, maybe some of them not. And actually all our endometrium is, there is some on drop-seq, but that was not part of human cell atlas. The majority of human cell atlas is single cell RNA-seq. The other question about the imaging, we are using different technologies for spatial. In 2D, we are using spatial transcriptomics, and for higher resolution single molecule FISH. Then for tissue clearing,
but how do
we
you
are
get the
still
3D?
Going for kind of 4-plex in tissue clearing, but we are exploring other options.
Okay, sounds great. So last, but certainly not least, is the breast.
Okay, so I'm Walid Khaled, so I'm the University of Cambridge and the Cambridge Stem Cell Institute. So I have 3 things to say: the why breast, and what have we done, and the data sets out there, and the challenges. So the breast is obviously a very dynamic tissue that changes quite dramatically throughout life. It is still the #1 malignancy, of course, breast cancer, and there is a need to understand cellular changes during normal and disease states. So in Cambridge, we have sequenced 180 samples from 75 individuals, which represents different ages, parity statuses, pre- and post-menopause, germline mutations, and actually also cells in milk, which we published earlier this year. These are all 10x preps, and we have about 1.1 million cells. And another group in the US, led by Nick Navin and Kai Kessenbrook, have about also 100 individuals, and they also do 10x, and they have a spatial data set as well. Other 2 data sets of note, Jean Brugui published one earlier this year as well, where they have also 10x data and CyTOF, predominantly lots of CyTOF data. And Jane Visveda also has 10x from normal and breast cancer samples. So the big challenges for us is integrating the data sets. So that's what we end up talking about most of the time, and I think we need to get some funding to get all these things together and have someone dedicated to put it together, because we think it's quite rich. And ideally we want to future-proof it, because even with the large numbers, I don't think it's large enough to see some of the changes that you would expect to identify in the tissue. Cell annotation is another one. Some labs have their, I'm sure this is a common thread across the conference. People are talking about the same issues. So that's it. Have I fit the 5 minutes?
Wow, yeah, that was under time. That was the shortest one. So what is the most challenge? I mean, we're going to dig into this question again with everyone, but since you have more minutes and you can sort of, spear this, what is the most challenging thing for you in the data integration? What's holding you back?
We basically don't have the human resource to do it at the moment. So I think we need someone dedicated simply to do it. So across our 3 labs, between Kai, Nick, and myself, we couldn't find none of us could take it on. So I think we're looking into ways of funding this, yeah.
Mm-hmm. But in terms of coordination between the 3 of you, you're doing well.
Yeah, I mean, we're one of the younger groups, networks, so unfortunately...
Out of curiosity, how many cells do you have?
I think between us about 2 million.
Oh.
So it's quite
that
big.
Was quick for being so young.
Yeah. Well, the problem is the network was formed during lockdown, so I think our first meeting was not a very happy one. So we haven't really progressed much.
So I want to get an idea of, what we have here in the room and who's who. So who here is, and you're allowed to, raise your hand more than once if you are, have multiple affiliations. Who's part of or identifies with the liver network? Where did Bruce go?
But Gary is joining
Gary's
online. Gary, hi.
Yeah.
Thanks
Okay,
for joining.
excellent. How many online people do we have right now?
I can't tell, but I want to ask the people who are joining remotely to please ask their questions by typing them, and I will read them to Donna.
Okay. And who identifies as lung? Oh, wow. Strong showing here. Who identifies as reproduction? Okay. Group there too. And breast. Okay, so, except for, well, our liver friends are online, so, and Gary's a good friend. So we're going to go through a series of questions, which I'm going to ask sort of each network, and I'm going to try and sort of change the order a bit, and so we won't go through the same order each time. And, they're going to be sort of with different themes. And I think it's really important to sort of both sort of try and get more quantitative, like, what is the nitty-gritty, what is the numbers, what are the technologies, what are the actual issues? And, I'd love for you guys to also share successes. What do you think you've managed to figure out that's valuable, that you think would be valuable for the other networks to know? So, we're going to start with a little bit of computation, and, again, we're going to take a few minutes for each network, even though I'm going to ask a loaded question, with 3 prongs. The loaded question is as follows. What are the most important, computational challenges that you have right now in your network? What are you trying to do to deal with them? And what do you think the AWG / computational network outside your bionetwork can do most to help you? So we are going to start with reproduction, and anyone from the group can answer. Just keep it short.
I'd be happy to share a couple of things that we've encountered. So one of the atlases that I work on is Human Ovary here at the University of Michigan, and we ran a spatial transcriptomics sample using the NanoString Genomics platform, and we've been working to integrate that data with our single cell samples and compare the two, and partly because NanoString Genomics is not heavily in use in the literature yet. We've it's been a learning curve to understand how to integrate that with single cell and do that robustly. So we're looking forward to seeing more groups use that technology and kind of develop a more standardized workflow for that.
So at this point, most of your integration efforts are within individual projects rather than across projects and labs?
We are doing both, Dana. So there are some projects. Actually, we got this, the Reproductive Tissue HCA award that is to integrate all of them. So we are now starting to integrate projects across labs.
Yeah, so again,
And actually that will be a challenge. And I think the other challenge building on that is not comparing things across kind of all the reproductive tissue, but also in other tissues, because there is a regenerative kind of cycle that will be interesting to also compare with other regeneration in other tissues without removing kind of variability. And again, with all this idea of gene programs, what gene programs are kind of shared between tissues, and that's kind of an interesting challenge to have.
Yeah, just to give you some feedback on your specific question, because there are some methods out there. Some of them were highlighted in Aviv's and Sarah's talk. We recently did, thank you so much. We recently did a benchmarking, and I can push my own method, Bayes Prism, that Elaine Fuchs just presented, if, that's my own method. In our benchmarking, Oli Stiegler's, I think it's cell to location works incredibly well as well, just to give, those are really good methods. As I said, Bayes Prism with a grain of salt, it's mine, but at least in our benchmarking, it's particularly good at getting the rare cell types, which is what I care about. So the next network I'm going to throw this question at is liver.
So I guess I will answer. So we have we have about 25 labs across 8 countries, and I think we have about 100 adult samples and a dozen pediatric samples and 20 fetal samples for version 1, hopefully by the end of this year. And so data integration is, I think we're able to do that. We've already done it with smaller sets, but we have it's still seems to be slow, and I think that's mostly because people are so busy doing all sorts of, many different analyses and many questions, and so I think there's
so there's no one dedicated on your team to do just the integration of it all?
Sorry, I didn't.
So there's no one dedicated on your team to do the integration of it all?
So we've had people dedicated to integrating within labs, but they're so busy with finishing those integration projects and pushing them forward that we haven't been able to integrate across. So I think that's the next phase for us, is getting the pipelines, all the integration pipelines sorted out within labs. People are testing them, validating them, and then applying them more broadly.
So, so in your view, what are you missing to make more progress? How can the computational community outside the liver network help you the best, or you just need more time or more people in the liver network?
Yeah, I think it's the latter, to really get that integrated map. Although there are specific challenges like the spatial transcriptomics point that was mentioned just now. Although, it's sort of like data overload right now, and we need to build all the systems. I mean, one of the things that might be good is for there to be more communication between computational groups. I'm sure there's a lot of reinventing the wheel everywhere. We're probably all implementing our own scripts and evaluating all of the, deconvolution methods ourselves. We also just evaluated a bunch and found Bayes Prism to be the best one for us for another project. So it would be great to have more ability to share experiences across the computational groups. We've tried to do that within the liver network. We have a monthly computational subgroup call, but even wider than that would be helpful, I think.
Yeah, originally I wanted to sort of share AWG benchmarking, and people felt that it would put us in a political shitstorm and that we should play nice with everyone rather than, but maybe, you know, the field really is looking for this, so we're just going to have to try and do this fair, and if we don't benchmark you well, it's not personal. So let's go on to the breast network. The question is, what is your big, not necessarily integration, but what is your biggest computational challenge, which seems to have a theme of integration, because we're going to have a separate question on integration. But yeah, what is your biggest computational challenge? What's holding you back? How can the AWG/broader computational community outside your network, how much of a computational community do you even have inside your network? You know, basically address the biggest computational challenge in your network,
So,
whether or not it's integration.
so I think I would struggle. I think it would be integration still, the main issue. So I think within our network, so this is done in collaboration with John Marioni. So he's involved quite heavily in this project. I think the typical cell annotation issue as well, which I think also is one that the breast field need to come across and decide on, calling things what they are. So the cell annotation, I would say, and the integration, but we haven't faced any other major issues other than that. Yeah, it's actually worked out fair. The experimental design at the start helped us. I think we sat down and really thought about it. So I think we eliminated a lot of potentially the batch effects and all of that.
Okay, and last but not least, you get to answer the question and also, you know, describe your experience with, you know, having someone like, you know, Fabian, Lisa, and Malte, you know, really engage with you guys, and how did that go, and what's still missing for you?
So, well, first of all, I will perhaps let Lisa answer. Just before, I can perhaps say that we benefited from the work that was done by Fabian's group regarding the test, the comparison of the different integration tools. For us, for instance, at the moment, there is a very good resource obtained with the SCVI, and that we couple with SCNV, and that was exactly what was proposed by Malte in his paper. So we are lucky in the sense that it was long that he has used, and so we can transfer directly into our lab. And so that's interesting
It's not entirely luck. Sasha was very charming towards the computational folks.
Mm-hmm. Yeah. I think that it's interesting because in our case, it's the direct comparison between COPD and control, and that helps us a lot for doing this comparison. But now, for the direct integration, I let Lisa answer.
Yeah, I think what we are looking at right now, so we already did the integration, and the way we approached it was we integrated all the healthy data, and also that was fairly homogeneous, let's say, in terms of technology, so it was all single cell 10x, and the rest of the data that we got, we mapped onto that using SE arches, this query to reference mapping technique. And I think the biggest question from a computational perspective we're trying to answer now is how well does this mapping work? We already looked into a few examples in detail that seem to work well, but we don't know exactly where the boundaries are in terms of how different types of platforms can you map, and how well does single nucleus mapping work? So that's kind of trying to figure out the limitations of this, let's say, because eventually we want to go through this in iterations, so new data sets will keep coming out, and we want to keep reintegrating those all together and then mapping new data sets using this query to reference. So we're kind of trying to figure out at what point this integration breaks and this mapping breaks and how we can resolve that. I think that's computationally the question right now.
Okay, we're going to revisit computation and integration, but, you know, I want to go now, you know, back to, again, we're writing roadmaps. Roadmap documents are due in the summer, and part of the roadmap is experimental design, it's the samples, it's the diversity, it's the metadata. So first of all, I'd like to bring my diversity colleague to the stage because, come on up. And you could participate in this discussion because, computationally, it's ideal if the computational folks are involved from the beginning, if the computational folks can help in the data design, but, there'll probably be more iterations of data collection, but right now, in terms of people's roadmaps, what are their plans? How and what type of diversity do they plan to include in each atlas? And how are you going to really, what technology, how you're really going to annotate the metadata, what technologies, what depth, those type of design questions for your roadmap. So let's start this time, let's start with lung this time. Oh, there's all the microphones are up here. We should have one in the audience. Okay.
So in our consortium, I need to check with my colleagues, but I think that there is a work on diverse ancestry at Sanger, but it's true that we have not a specific point. For instance, in Nice, we have only the access to the local population. It's probably similar in Groningen. So I would say that we have a limited diversity at that point. For our colleagues in Chicago and in the United States, probably there will be a larger diversity. But,
so this is racial diversity you're talking about.
Yeah.
But there's other issues other than racial diversity. I know that there were discussions about trying to get things such as smoking status, altitude, and other really important things for the lung. So this is diversity in all its aspects, not just in the, politically correct, do we have all races included, which would be fabulous because previous projects neglected this. But now we're talking about the experimental design in the whole scheme. What other components did you manage to include, and what type of metadata and scope and scale do you attribute to trying to capture human diversity in all its aspects?
The other important aspect that was considered was the age, because you know that the lung is probably one of the most affected tissue with regard to age. So and again, we are not working with thousands of different samples. We are working with tens of samples, and I think that we will have soon.
Why Lisa's atlas already has 400, or how many healthy do you have?
116.
Oh, 116. Okay. So, and the and how did you handle the metadata so that Lisa's life will be better?
Yeah, that was a painful process. We tried, basically we set up a table with standardized nomenclature and asked people to fill it out, or we tried to recover the information from papers. But that was actually what took us months to get together. And so the metadata in terms of demographics we collected were BMI, age, ethnicity, sex, and that's it actually.
Yeah, so again, guys, metadata in advance saves a lot for all the networks. So let's do breast next.
Yeah, I think for the metadata, that was essential for us from the start, especially if you're going to look at effects of parity and age, at least you need to have that information. So we actually got most of our samples from a tissue bank in London called Breast Cancer Now. And the reason why we went with them is that they had 80 data points that they could give us on each sample, metadata points. So that was one of the main reasons why we used that.
And do you have diversity? I mean, London is fortunately a very diverse city, but not a diverse population would likely be, into giving their samples there.
Yeah, so I don't think we'll probably capture diversity. I think you're interested in that.
Yeah,
you're doing some of that.
Next, yeah.
So yeah, so I think that we really spent a lot of time collating the metadata up front, and we rejected any samples that didn't have the minimum amount of information, because otherwise it would have been of a waste. Yeah.
I mean, we have 5,000 normal breast tissue sample. All of them are genetic ancestry mapped. These are FFPE and frozen. Limited number of them are from the cryopreserved. So these are taken from healthy women from a specific location. So we are just making a standardized technique. And at the moment, our lab has generated 20 Ashkenazi Jewish, 20 European ancestry, 10 Native American, and about 5 or 10 each of Asian and his Latina, as well as the African American. These are multi-omic assay data. They are, most of it is the CZI cell by gene now. It will be soon transferred to the HCA network. So there is more data coming from our group on that.
Yeah, that's, so what technology did you use since it's
All 10x genomic multi-omic. Initial work was the single cell that is already on the HCA, and then now is the multi-omic.
Okay, and liver?
Oh, Donna, we have a comment from the lung coordinator, Martin, who's joining online, and he just mentions that for lung diversity, we're now increasing diversity by very actively involving HCA Asia and HCA Latin America, as well as HCA Africa.
Oh, that's exciting news. And there's success in actual sample accrual? Yeah.
Oh, someone can answer on his behalf?
So yeah, since we actively started looking for this, we have a few more data sets that are way more diverse. So I would say probably 30 or 40 extra individuals that are not of European ancestry, let's say. So yeah, I don't know to what extent that is a success, but it's an improvement.
Okay.
Yeah, I just wanted to add, so we were funded to collect cases on breast, fallopian tube, and prostate. And for us, I think it's going to be 80 women in 6 spaces, so Miami, Kenya, Nigeria, Haiti, Jamaica, and the Bahamas. And we're capturing germline genetics at the same time as, of course, like Ancestry. So that will be, we just started 6 months ago, so we're in the process.
Okay, liver?
Oh, can we bring Gary on, please, via Zoom? And Bruce, if you want to chime in.
Yeah. Okay.
So, can you hear me?
Yes.
So for adults, because we started a few years ago, we're collecting metadata, but it's not totally standardized and complete across all the groups. We have very good diversity for sex and age, and sort of incidental levels of diversity for genetic ancestry. So there definitely genetic ancestry is represented, but it wasn't specifically targeted. For the more recent started pediatric liver project, we're planning this more rigorously up front. Alan Mullen is the other liver coordinator who's helping lead that in collaboration with CZI, who I think it's great that HCA and CZI are, and just even here are making sure everyone's aware of thinking about diversity through all the people and all the way up and down the team, so that everybody can think about that when they're doing their work. The big challenge for pediatric samples, because liver samples only come from rare medical cases, are that they are often easy to identify. So there's a lot of discussion right now about how to deal with that. 2 other points I wanted to make quickly. One is that establishing single nuke RNA-seq profiles from frozen tissues really enables us to do better experimental design, because especially if we have freezers full of samples, we can go to those freezers and select samples based on different factors that we're interested in, like genetic diversity, and rather than just waiting for samples to come in from the operating room incidentally. So that really helps. But it does require that technical effort to handle frozen samples properly and get the best data from that. And then I just had one question that I wonder if we could discuss at some point. I'm wondering if anybody's tried to identify genetic ancestry by looking at Snips that are visible in 10x Genomics 3' single-cell RNA-seq data.
That's definitely a great idea. Unfortunately, there's a lot more identification in these samples than, you know, my own institute thinks that, like, they have a lot of restrictions on DNA-seq because that's DNA and that's identifiable, but they haven't quite figured out that you can figure out a lot from RNA-seq as well. Fortunately, these are lawyers, so I'm using the their ignorance to my my advantage. But yes, is anyone in?
Yeah, I could comment also. So we're trying to do this right now in collaboration with Joseph Powell, and I think there are even a few other groups that are simultaneously working on developing a method like this. There are also people from CZI who are better informed about this.
Yeah, we really need to do a better job at sharing with everyone methods that are well benchmarked, both methods that are, you know, pretty bespoke like that, as well as something like the spatial transcriptomics deconvolution, which everyone is asking for. That's I guess on me. You know, reproduction. Last but not least, for this round.
Yeah, so far we know that we have a diverse collection, and actually we have been looking at these SNPs, and that's why we know also that we have diversity in our kind of data set, maybe around 20 to 30% that are not European. But then now there are projects that are actually focused on this diversity in both the endometrium and the ovaries. Those are happening in UK and States, but there is also some efforts that we are aware on collecting placentas in Uganda, for example. So moving forward, that's something that we are considering.
So before we move on to the next topic, do any of you have something to add to the, you know, wisdom, how to get people engaged, how to get these samples, critical wish lists, sort of? Any of you want to?
I mean, I just want to make one comment. In our case, whenever we collect the tissues, we also collect the blood from those donors for the genetic ancestry mapping so that we are not dependent on the single-cell RNA-seq data to map them. So that may be one thing everyone can adopt at the same time you collect the blood sample. With respect to liver, I have a question. Are you recording any maternal as well as individual alcohol intake as a diverse aspect? The maternal alcohol intake as well as the individual alcohol intake as a diversity aspect in liver?
We collect a lot of information about the individual, including alcohol intake and you know, viral infection and high-risk drug use, all sorts of things like that. I haven't seen maternal, so I'm going to go and ask our surgeons and clinicians to see how that information is collected, and also specifically for the pediatric, I guess.
So I could answer some questions about, well, you asked about engagements in populations that are not necessarily represented. So CZI has thankfully funded the Ancestry network that clearly is based on majority non-European populations, and so that has allowed at least our group to be able to do single-cell mapping in populations that are actually highly represented in the US, but going to the locations to collect these cases. And so one of the things that we are doing, and I think it's actually required in many of the Ancestry networks, is community outreach and engagement. So not just outreach in terms of collecting and encouraging people to participate, but engaging the community to explain to them why it's important to participate in research. And again, doing a lot of public-facing activities. That actually for us, it's based in Jamaica and I think in Miami and in Kenya. And I think lastly, one of the things that we would like to do and other sites is geography. So where are the people from? Even if they're in the US or in the UK, there might be immigrants. So we capture that type of information, how long they've been in the country that they are in.
So for me, geography is, you know, it should be more than that because, you know, certainly rural, city
Yes.
levels of also pollution, all these sort of environmental factors that, there's who your ancestors are and where you live and what are the environmental impacts of that. And I think
Yeah,
that is
especially for the lung
incredibly
people.
valuable metadata for investigation going forward. So thanks very much. We're now going to move on to the charged topic of cell annotation. And again,
There's one question, Donna.
oh, question, yeah.
Can I ask a potentially very naive question? So I've mostly worked with bulk data myself with large cohorts, and there
Can you introduce yourself real quick?
Sorry, Rob Terhorst from SEM. So there we always do a sex check and an ancestry check in the sequencing data, so RNA-seq, ATAC-seq, to make sure there's no sample swaps. Is this also for single cell a typical workflow to always do these checks to make sure that no samples were swapped compared to the annotations you have for them?
I don't know if Lisa did that. It's not that typical, I think, mostly because we haven't really included those aspects too deeply in most of the atlases, but again, lung is like ahead of the game. So Lisa, did you guys do that?
Yeah, well, we did it later on when I noticed that there were a few women with Y chromosomal expression, and so then we figured out there were a few samples that were probably mixed up, but it's not something we standardized or like.
It should be.
For me, I've experienced this a lot in
lab
build data.
There are lab
that's a recommendation.
That's a good point. So cell annotation is a big charged thing. I know, again, most of my experience is also from the Lung Atlas, where a lot of the people definitely didn't reach consensus. People call different cells by different names. There's a lot of undiscovered cell types. Some systems are more advanced than others and even developing an ontology. So my question to everyone is, how well are you along the line? How are you coalescing as a community to reach consensus cell annotations? Through what mechanisms and collaborative efforts? How are you balancing, the sort of statistical input that things such as clusters and the data gives you versus, your favorite markers to really reach at a consensus data-driven, biology-faithful, first pass cell annotation? And I think I'm going to start with the lung again because they really did that. Kudos to you guys successfully, so you can share your experience and say what's left. And I know that, Pascal had a great idea that COVID destroyed, but yeah, share your annotation efforts and challenges with the group. And then everyone else can share their experiences.
Yeah, I can start, but I'm not necessarily the person
Yeah,
who
or
contributed
Pascal
most
can
of
do it as well.
Yeah.
I think you led that charge, right?
I can explain the system we used. Let's say, so the way we dealt with things is first we used the annotations from the individual data sets that people provided to us, and we mapped those to sort of a common terminology. And wherever we weren't sure if things overlapped, we just kept them separate. Then we integrated everything, and then we found out that there were a lot of contradictory annotations between groups. So we actually re-annotated, and we asked 6 different independent, let's say, experts to completely annotate the atlases to the best of their knowledge, let's say, and from there we tried to reach some type of consensus annotation, and in some cases people weren't sure or people contradicted each other, so in some of those cases we just left it at a coarse annotation, and in some we just made a call where it seemed like the field just didn't agree in general in terms of where one cell type stops and the other starts. So.
Yeah, but Pascal, you made some efforts to reach consensus with your colleagues in a sort of collaborative setting.
Yeah, so it is coming to the idea that I was saying at the beginning, meaning that it's really a community effort and not from the single cell only, but really from the lung community. And so, well, unfortunately there was COVID, but we were planning a meeting in Nice where people, experts from the community would share their knowledge about the definition of the different cells they know. In fact, this effort has been existing anyway, and there are, for instance, a very nice paper this year from the LangMap Consortium describing the different cell types. This is just a starting point, but I think that if you make a sort of cell card for any cells that you have, and that everybody can contradict yourself and say, well, I don't agree. I mean, it's a good point, and I think that this is exactly what we need to do for any cell type, and make by this sort of definition, minimal definition of the minimal number of cells that everybody agree, and then accept progressively to extend the collection. And I think that it is an ongoing effort that will take probably a long time, but it's good.
And one more thing for Lisa before we go forward. Can you share with everyone, the role and the importance of the cell annotations in your integration efforts?
Yeah, this was, I hope I understand your question correctly. So, but this...
Did having a consensus cell annotation matter for your integration, and how?
This re-annotation, let's say, you mean?
What?
You mean this re-annotation that
yeah.
we did? Yes. No, that was actually very important. So we did already a few downstream analyses before we did this re-annotation. And then after we re-annotated, we re-ran those analyses. And then we saw actually that our signals were a lot stronger, because before there was just a lot of mixing of what we thought was the same cell type, but were actually different cell types. So the re-annotation was really actually quite important in sort of strengthening the signal of downstream analyses we did.
So how does each network really deal with cell type ontology, nomenclature, and annotation at the network level? Let's go with reproductive next.
Yeah, so I mean, actually for the reproductive has been very challenging because we know very little about the tissue. At the moment we are just communicating with each other, and we need to do, like, in a way, organize better. But one of the things that has been super helpful has been the different modalities. So the spatial data to be able to locate cells in the tissue and kind of understanding better the attack for the transcription factors. And I can also let Andrea tell about other experience on how annotating data.
Yeah, I would agree with everything Roser said. And I'd also highlight that because our different groups are collecting samples from different ages, and especially our reproductive system that we're studying is so dynamic across age, we have to acknowledge a lot of the differences that are age-related among our samples. But with us as well, multimodal analysis and using our spatial data set alongside single cell has been great to kind of validate. And we've also used a lot of data sets from other atlases, other organ systems, to understand especially stromal cells in our data sets, because there's just not a lot out there about them.
So how much do you work?
I think moving forward, as we are starting to integrate data sets, we are starting to build these ontologies where we will be able, and we are still under discussion if we should be using markers of the cells, position of the cells, to kind of create this dictionary of cells.
But how much are you working in how to reach consensus as an atlas versus individual projects, and how much are you bringing in some computational input into that?
Yeah, so as I said, we are just starting, Dana, to integrate, because the reproductive system maybe has been more delayed than other systems. And at the moment, yeah, we are integrating data sets, and we are starting to face these and kind of generating these. I mean, there is actually, we have done quite a lot of exercise between data sets, and there is quite a lot of agreement. So maybe the thing that we are is how to name these cells, which at the end of the day is just a thing of nomenclature. But we do see a lot of consistencies between data sets, and we are in constant communication between each other.
Breast?
Yeah, I mean, we're at the moment just communicating, between the different groups. I mean, it sounds like we need to communicate more with others. And generally speaking, I guess because we have the epithelial cells and the immune and fibroblasts, so I think we rely on for the immune and fibroblasts, we rely on other published data sets to help us with annotation. But the two big groups of epithelial cells that are easy to annotate. I think once you get into the finer details, that's where it can get controversial.
I mean, the epithelial cell type, particularly one population called luminal progenitor, the nomenclature has been changing from publication to publication. Now we no longer call it as luminal progenitor as of last month. So the new name has been given to those cells by Joanne Bridges' group. So I'm not sure we have any consensus on what cell type, even within the epithelial cell type, what to call them yet.
Okay, and last but not least, liver.
So we've been fortunate in that the interests of all the different groups that are participating are very complementary in terms of the cell types that their cell types interest. So we have groups that are really specializing on T cells and macrophages and stellate cells and trying to subtype all of those. So we've been, we haven't had a lot of controversy, more like collaboration. When someone has a macrophage question, they go and talk to Martin Williams, or when someone has a T cell question, they talk to Georg Glauer. And
are these definitions?
We do have one challenge, though, that with some of the tissue-resident cells, that we need to distinguish between circulating and tissue-resident cells, and so that's a good reason for us to do single-cell RNA-seq on blood for some match samples to better understand which, where the immune cells are coming from, and that's important in the liver because blood is so, it processes blood all the time. It's really, like the blood-liver connection is really strong.
So my question to you is, again, is there consensus and collaboration? So the macrophage expert that you just described is his or her method of annotation used across the network, or different data sets are annotated really, really well for for the lab's favorite cell type, and don't trust the other annotation. So is there at the cross group bionetwork level sort of consensus and shared knowledge about the annotations?
Yeah, we're sharing a lot. We share markers, and people look at each other's samples as they're coming online. We haven't formalized this and standardized that yet. We've been in touch with the HubMap cell type tables and Kati Borner, for instance, to offer collaboration to help define liver cell types and standardize them. But I think there's still a lot of subtypes being discovered right now. So every time we look at a bigger set of samples, we get more subtypes, and so it's I think it's going to take. We're in a phase where we're discovering a lot of subtypes right now, and we need to get through that phase and standardize those, characterize them better.
All right, so let's go to integration.
If I may just add a point, I think that we are arriving now for the lung at a level where it would be also interesting for us to complement or to discuss with other network, for instance, the immune or the endothelial or any people working on fibroblasts, because I'm sure that, and also development naturally, because there will be a lot of information that will be complementary to what we have going.
Yeah, I hope we'll have time to discuss plans for potential cross-network activities. That's sort of on my forever agenda that I'm supposed to squeeze in. Where am I for time?
We have 20 minutes left, and please remember to spend about 5 minutes summarizing, because you will be reporting back.
Yeah, I know. So, we'll try and do the next one really quickly, but we talked about integration. So if any group can really crystallize and clarify, not just integration is hard, but can you pinpoint your biggest pain point in integration? This time, we'll start with Gary, because, we're trying to mix the order up. So you were last time, first this time. What's your biggest pain point? Is Gary still there?
I guess the biggest pain point is really just, as I mentioned earlier, having someone who's dedicated. Right now, our best idea is that we're going to have all the computational biologists come together to make a big big integrated, make the map paper, basically. And we have a lot of people interested, but pretty busy with getting all of their, work done. So that's, I think it's going to happen, and we should be able to handle it. We know how to do it, I think, because we've done it a few times on smaller data sets.
Okay, Breast, what's your biggest pain point?
Yeah, I mean, I already mentioned this, is it's just having the dedicated time to integrate all these different data sets. And actually also, we need to, as a network, we haven't actually really come together yet with all the different groups. I think there needs to be more of a reach out to the other groups.
How many computational people do you have embedded in your network?
I mean, I don't know about the other groups, but I mean, for the Cambridge effort, we have 3 people just working on that, on the data set itself. But I don't know about your different networks, yeah.
I have only half bioinformatics person integrating our data, so I don't think we have much of a communication with the other 2 groups in the US regarding data integration at the moment.
Mm-hmm. And reproductive?
Yeah, so we are integrating data, and I think one of the most challenging for us is to avoid masking the biology, because there is this wave of regeneration, and usually we pick them from different kind of individuals, so we just need to be very careful with that.
So I didn't quite understand. So what exactly is what is the most challenging part?
Yeah, so we have a lot of confounding factors, because we have these regeneration cycles driven by the hormones, and usually you don't pick from the same individual 2 different stages. And due to this variability, and that at the moment we don't have hundreds of samples, we will have, that's a challenge that we are facing for the integration. But we are working with these, and yeah.
How many computational people do you guys have helping you in the network?
So, I mean, we have several, and actually we have just got this grant from the CCI to focus on this integration of the data and the database. At the moment in my team, there will be like 3 or 4 people. So, yeah, I think we are quite fit with this.
And Long, what's your biggest pain point at this point?
Yeah, for integration, I think the hardest part was to actually assess how good it was, if it was good enough. And that was basically what was all the downstream analysis to figure out. Are we able to find rare cell types? Are we able to see variation between individuals? Can we explain the outliers, or is that batch effect that's still remaining? I think that was what actually took a lot of time. And then I think the current, let's say, biggest imperfection of this atlas is probably that we're not able to, or so far we haven't been able to really see subtypes of T cells very well. So it seems that that cell type in particular might have over-integrated, or we need better data or better annotations, something there that could be improved.
Yeah. I think that there is one point that we have not discussed so far. Our atlases are mostly 10x 3' or 5' in the best case. But so we are speaking always of the same approach. I suspect that there will be a diversification in the coming years. And so that will probably create new situations. And I'm not saying that it is a real problem, but we have to consider it.
Oh yeah, no, the other modalities are definitely a big challenge. I think one of the one of the issues is that we have collected so much, 10x 3' data, and even more generally, single-cell RNA-seq, whether it's in-drops or drop-seq or 5' data or whatnot. And I think, for me, yes, we need to diversify in multiome and attack and spatial transcriptomics and MERFISH and 3D tissue clearing. And of course, as a computational biologist, that interests me the most. But given the amount of data that we have collected, which is, dozens of samples in the smallest atlases and hundreds of samples in the largest atlases, I think the community is waiting us to show value from the large, show the added value of the large RNA-seq. And, added value and, what Lisa just said, how how, does anyone have ideas of how the networks could collaborate, for example, to get a better understanding of the immune cells? Pascal also mentioned the endothelial cells. There's a lot of shared cell types across tissues. And really, what type of things do you suggest in terms of cross-community efforts? What would excite you? And if anyone can give like a more crystallized, more concrete idea, then let's just compare the immune subsets. That would be great.
Yeah, thank you. We work on the Hooter program, the human uterus, and your question is very important for us because we realize how important is the immune system. I mean, we knew that, but not only for embryonic implantation, but also for every uterine pathology. And for the stem cell niche that we are very, for us is very crucial. What the key lecture today, it was very revealing in what could be the impact of the lymphocytes and the immune system in what we are doing. So I think that this cross-communication with the immune network, Cell Atlas, will be very important for us to really advance in the knowledge, because we are not experts in immunology, and the immunology is completely rewritten in our organ.
And how would you suggest, like what ideal ways would you like to interact with the immune network?
Well, I already spoke with the coordinator, asking her exactly that. How can we get together, bring the data, they can give us the feedback, and so we can be better off on that.
Not to diss my colleagues, because they're, super amazing, and I think, Chloe is a queen, but I will say that immunologists are a lot better at, cells in immune organs, and actually, tissue-resident immune cells are a challenge that the immunologists are now really embracing. But keep in mind that immune cells, once they enter the tissue, are behaving very, very differently. And I think if the immunologist had, and I'm going to put words in their mouth, but I collaborate with enough of them that I feel semi-comfortable doing this. If there's one thing that really surprised the immunologist from all these single-cell studies, is just how different, how diverse, and how transformed these immune cells are when they enter the tissue. And I think it would be fascinating to see how, we're always thinking about how the immune cells impact the tissue. I'm interested in in the other way around, how the tissue impacts the immune states.
Yeah, no, fully agree. And the best example is natural killer cells in blood or in the uterus. But at least if we can have their view, I think we will learn altogether more than that we are doing now, because this is a complete new animal now.
I totally agree with this, and I'm just wondering if it wouldn't be an opportunity for an inter-network, sort of effort that we could look at tissue-resident immune cells, and you could even divide it by the myeloids versus the lymphoids, and those are pretty separate universes, even though they crosstalk a lot. But I mean, really, there's so much that you compare lymph node and spleen and bone marrow immune cell states to tissue resident, way different. You compare that to PBMC, no way. It's just you must really concentrate. But I think there should be there will be convergence, and higher resolution that no individual group could achieve by
yeah, of course. Immune cell cell types in tissues is on the list of interesting things. The problem is that we have too much to do and too little time, money, people, FTEs. I mean, I will say that what you just suggested. We got a first very beautiful and very well done pilot by Sarah's group on comparing cross-tissue immune cells, which was published in that. There was like a first tranche of, I actually think, very well done, and I'm a critical person in personality, cross-tissue atlases. But as we get more data, more tissues, more networks, more people engaged, and particularly the scale of data that we're talking about now, I think we can dive back in there. So, trying to again hone in, thinking of what was already published, if any of you read, some of the papers that were just published. Also, Aviv saw some really beautiful cross-tissue behaviors, again, of macrophages that was, incredible. Macrophages, I mean, those guys really, I think they're one of the most plastic cell types. What can we do more other than, that first tranche of paper in Science, if anyone sort of read it and got ideas of, well, why didn't they do this?
Mine is a biologist asking a computational suggestion, giving a computational suggestion. As the multiome assay data is becoming more common now, and assuming that the ATAC-seq is much more stable data than compared to the RNA-seq, should there be a working group to integrate the ATAC-seq data for the cell type annotation across the networks?
Yeah, so, both, good integration of ATAC and RNA and getting good information out of ATAC is actually one of the biggest challenges in the computational biology community. In terms of, combined atlasing questions at the level of data integration and at the level of cross-tissue efforts, I think we just don't have the critical mass of samples yet. I'm really hoping that in the current roadmaps, all of you are planning to collect a lot of multi-ohm data. I see we have 10X sitting in the background, cranking a yay, all this money is going to come. I asked 10X, please reduce the cost of your multi-ohm. They're really ridiculous and holding us back. And if you reduce the price, you will actually make more profit because we will do much more of it. We just have limited budget, and multi-ohm is really important for our efforts. But right now, we don't have enough of it. So. But that's, there's so much we can do once the critical mass of multi-ohm accumulates in our community, and nothing would excite me more. All right, I'm supposed to report back, and this was a great discussion, and I hope it stimulated a lot of the bio-networks, and people could sort of say, hey, we're not the only ones struggling with integration, we're not outliers, as the only ones that are doing pretty good are Lung, and that's because they have Fabian, Malta, and Lisa, and we're going to try and replicate that. Maybe, Lisa, do you want to be cloned?
From Lisa. A stem cell donation from Lisa would be a good approach.
But what do we want to share as a group with a bigger group now that we're going to bring like 5 of these little breakout groups together? Is there anything that you think is important that I can, wing it, but I want to know if in this group there's important things that you want me to relay to everyone else?
Maybe I'd like to emphasize the fact that the cross-tissue integration can really answer very basic science questions. We have talked about immune, we are interested into epithelia, and there are some very basic questions that have been, looked for years, and having very similar cell types, and we have heard in the keynote today about gut and skin, and there are many other examples where very similar cell types are across very different tissues and having similar functions but slight differences. And in developmental biology, we have wondered a lot about that. And I really think that this comparison for very similar cell types in different tissues can help answer very, not regenerative medicine questions, but very, very basic science questions.
Okay, so thanks everyone for wait, is there any other point people want to make? Okay, I think everyone is, we have a coffee break now, right?
We do. So actually, the report back starts at 6, goes till 6:40, at which point we will close and walk to the restaurant together. But the session can potentially go till 5:45. We have another 10 minutes, unless you want to let everyone
Is there
out early.
again, I just, I don't think we have time to open another full topic because there's 4 bio-networks, and unless there's burning questions here in the group that people want to discuss, I'm happy to discuss them.
Kai, can I ask a question? Like, as a user, I've noticed that only in the liver atlas the pediatric samples were mentioned, and I personally work on pediatric sarcomas, so for me, I would be super happy to integrate into a pediatric cell atlas. And is it mostly ethical limitations and it's just harder to get approval, or it's just never going to happen, or it's the next step in the future?
It's a combination of a couple things of ethical limitations, even though, again, this varies by country. So to get both genetic diversity and pediatric, for example, will be a challenge. I think it's a little bit about the history, both when people came together and the initial, I mean, we wanted it all from day one because we're greedy. But when, we were talking about these ideas with funders when this thing launched in 2016, it was like, oh, no, don't touch disease, don't touch pediatrics, don't touch diversity, just go after a deep cell consensus. Like the big, rave then was, get the rare cell types. What is the power to get rare cell types? How do we know if we're missing rare cell types? And it was very much, forget diversity, forget disease, forget pediatrics. Because I think of the success of just the biology that's coming out of these technologies and the excitement and how quickly we managed to galvanize together as a community. I think what makes the HCA special relative to some other scientific endeavors I've been involved in is the community, is the fact that we are collaborative, we do share experience, we do come together and share rather than compete. And I think that's also both helping making our science so awesome and helping the funders like us, I think, I hope, I don't know. But yeah, so for example, in November, there's going to be, and if Gary is still online, he's one of the main organizers of this. There's going to be a meeting around Developmental Atlas and Pediatric Atlas, and it's a late bloomer because suddenly there was more openness to fund this, but not because, like, for me, development is my favorite question. First, really, I want to go to the fetus, but even, pediatric. And yeah, pediatric is certainly more challenging. Gary, do you want to add about the pediatric atlas, not just for the liver, but, a bigger effort across the HCA and your planned meeting in Paris, if you're still there?
Yeah. Thank you for advertising a meeting in November 20th to 22 in Paris. The first meeting was last August. You can actually go and look at the recorded lectures on YouTube. There's a lot of interesting discussion to fill in what all the different activities are in pediatrics, and there are many. And the ethics questions are resolvable. We have a really good connection with ethics right now with Bartha Knopfers and others. So they had a great panel at the last pediatrics meeting that discussed how to resolve a lot of the ethics issues that we might be facing. So yeah, let's continue the discussion in Paris in November.
Yeah, so you should try and come to that one as well.
I just want to make 2 points. One, it looks like the genetic ancestry or diversity is still in an infancy state. It's not. Oh
yeah, that is even more infant than pediatrics.
Right.
You guys joined the fold, but you guys joined the fold, with full force funding behind you. So that's helpful to join the fold this way. I mean, there's what we want to do, and we're a community. We're not funders.
Right.
So we have to convince, and part of the reason that we have funders is again, people on the OC kept on telling the funders, diversity, important, we can't make the European male human cell atlas again.
Yeah. Second point, which is becoming more common with the biology, is the distribution of the adaptive traits, like, for example, Duffy, sickle cell, those aspects having an influence on the normal biology. And the percentage of people with sickle cell is much higher, and we are not taking that into consideration when we are generating this map. Maybe the next step after the genetic diversity is on the adaptive traits, which are distributed because of the malaria infection, whatever it is in the population.
Okay, any other?
We do not have any questions from the virtual audience, but this is your chance. There's a slight lag with how we're broadcast, so please give it a couple minutes just in case someone comes through virtually. But otherwise, we can probably wrap up.
We could probably break out and go to the porch before everyone grabs our seats and breathe some wonderful fresh air on the porch, which should be, well, actually, it's probably still brutally hot.
Yes, so we will see you at 6 for a report back. It will go from 6 to 6:40. Thank you, everyone. And Donna, thank you so much for beautiful moderation today.